Hello everyone, I'm Dr. Desin. Welcome to this session on introductory microbiology. Microbiology generally involves the study of living things which are really small, which are difficult to see with the naked eye, and in many cases we'll have to use a microscope so that we can magnify what we're seeing. Some of these are of course harmful microbes, others are beneficial, and we'll come across a lot of these as we talk about microbes. In terms of microbes, we are going to mainly see microbes that are eukaryotes, prokaryotes, or viruses. Within the eukaryotes, this includes protozoa, algae, fungi, helminths, or worms. When we talk about prokaryotes, we're mainly talking about bacteria. And viruses, we talk a lot about viruses. I'm sure you've heard a lot about viruses ever since COVID. These are acellular and are some of the smallest microorganisms that are known. And then we have organisms, or not organisms, but agents, infectious agents, which are not really microbes, but they can cause disease like prions, made of proteins, can cause severe disease like BSE, mad cow disease. And we have, of course, viroids, which don't really infect us, but infect plants. So the main type of microorganisms we're going to see in microbiology are the bacteria, fungi, viruses, protozoa, algae, and helminths or worms. When we talk about microbes, the basic structures of organisms, or some of the organisms we're going to study include prokaryotic cells. Eukaryotic cells, will compare and contrast these a lot, and then of course viruses. A big question that often comes up in the study of microbiology is how small is small? Because we're talking about microorganisms, but of course it's all relative. So when we talk about bacteria, we're usually talking about we usually refer to them in the micrometer scale. So most bacteria will lie between 1 to 10 micrometers in size. And with viruses, they're even smaller, if that wasn't small enough for you. And we're going to be talking about the nanometer range, and most viruses are between 10 to 200 nanometers in size. Microbes are found everywhere. They're ubiquitous. They're found in extreme environments, in regular environments, in salty environments, in the Arctic, in the deserts, in space, everywhere you go. In the clouds, in the air, etc. And we'll see many examples as we talk about microbes. Microbes are involved in an important process which involves energy flow. So some examples could be the decomposers. Anytime something is dead, whether it's plants, leaves, humans, animals, there are certain microbes like fungal pathogens or fungal organisms which are right on top of that. They decompose and recycle a lot of the material. They're important to us. There are other microorganisms that are involved in photosynthesis, others in soil formation. They make it better for us. We use microbes in the production of foods, for nutrients, drugs, vaccines. Microbes play a big role in bioremediation and oil spills. And microbes, of course, are involved in causing disease, and that's really the part which we'll look at later on. An important concept or term that often comes up in microbiology is the burden of infectious disease, and this is referred to often in terms of mortality and morbidity. Mortality refers to the death, the number of people that are killed as a result of the pathogen, and morbidity refers to the number of people that get ill. And this is important because it can range from anywhere from a few people to thousands to hundreds of thousands to millions. And if we look at what is killing a lot of people around the world, this can range from things that are not associated with microbes like diabetes, hypertensive heart disease, and it can involve other things like lower respiratory tract infections, upper respiratory tract infections, kidney disease, etc. Within the field of microbiology, it's really important to appreciate and understand the understand how this came into play and who were some of the pioneers who really started thinking along the lines of microbes and discovered a lot of these things. So we'll spend a few moments highlighting some of the pioneers that really brought us to where we are today. It all started off with this concept of biogenesis and spontaneous generation, or biogenesis versus abiogenesis. Like, do living things come about from other living things, or is it just spontaneous and they come out of nowhere? Some of these early discussions also led to the development of culture techniques, sterilization techniques, the germ theory of disease, etc. Some of the really important people include Antony van Leeuwenhoek. He really played an important role in the development of the microscope. As they say, seeing is believing, and he started observing a few things by developing a simple model of a microscope, and it was at the time referred to as animalcules. Then we had some important experiments by Francesco Redi, where he did an experiment where he had a jar with some meat in it covered with some fine gauze, and some flies laid some eggs on top of the gauze without having access to the meat, and then their offspring were generated as a result of that. And then at the same time, he did another experiment without the gauze. The flies had access to the meat, they laid eggs on the meat, and offspring were generated. So this indicated that the offspring, the maggots, they're generated from flies, or flies give rise to maggots, and it's not that the meat gives rise to the maggots, etc. That was one of the initial experiments supporting the theory of biogenesis, that living things come from living things. We also had a famous experiment by Louis Jablois, where he essentially had some infusion or some broth. It was boiled, it was covered, and as it was kept, nothing really grew in the broth. However, when it was sterilized and the cover removed, because it had access to the air and some dust, after a while they started seeing microbial growth in the broth. This sort of indicated that there are some microbes in the air which have access to this, and this gives rise to microbial growth. Other experiments include the Schultz and Schwann experiment, where they had 2 flasks with sterile broth and an inlet of air which was being sterilized, heated, and as a result there was no contamination, suggesting that microbial growth happens due to excess from the air, where microbes from the air can contaminate the broth and result in growth. We also had the famous Louis Pasteur experiment, where he had a flask with a swan neck, the swan experiment, and sterile broth in there. It was heated, and for an extended period of time there was no contamination. However, when the flask was broken and there was access to the broth, there was contamination. All of these things sort of supported the theory of biogenesis. Louis Pasteur, very famous French chemist and microbiologist, he showed that microbes could cause fermentation. He also showed that microbes cause spoilage. He disproved spontaneous generation of microbes. And he also played a role in developing the rabies vaccine. Robert Koch, very important scientist of the time, he really was instrumental, along with his colleagues, in developing the germ theory of disease. Really important, and if we think about it, it's so important when a new pathogen comes on the scene, and we really need to show and prove that that pathogen is causing the disease. So it basically states, this theory of disease basically states that germs or microorganisms cause disease, and it's not by sin or bad character or some omen or poverty. And the 4 Koch's postulates are really important. In any microbiology course, you really want to know, you really want to make sure that you understand the 4 postulates, Robert Koch's 4 postulates. The first one being that microorganism which you think is causing disease must be found in all animals or beings suffering from that disease. That's the first one. So you shouldn't find it in healthy animals or subjects, but you should find it in those that have disease. Okay. And then step 2, you should be able to isolate it. So let's say I have a mouse that is ill. I should be able to isolate that organism, which I think is causing the disease, from the mouse and grow it out. Okay, that's step 2. And in step 3, I should be able to take the organism on my plate or my culture or my media, and then when I introduce it into a healthy subject, mouse, animal, human, that person should get that disease, or that animal, human, subject should get that disease. And lastly, number 4, when that animal, human, or subject gets the disease, I should be able to re-isolate it again. With these 4 steps, you can definitely, to some extent, prove that a disease is caused by a certain pathogen. It's not just some random thing. Other important contributors to microbiology include John Tyndall. He was instrumental in developing this Tyndallization technique. It's a type of sterilization technique, not so commonly used today, but still exists in some parts. Joseph Lister, he developed aseptical techniques, or played a major role in developing aseptic techniques, especially in surgery, etc. Then, of course, there's Edward Jenner, also referred to as the father of immunology. He played a huge role in really establishing the procedure for vaccines. And there's an amazing story out there, very nice diagrams out there. You'll find a lot of microbiology texts which show really how he developed the smallpox vaccine somewhere around 1796. As microbes were being discovered, it was important to have a process for classification, for taxonomy, and one of the persons who, or microbiologists or scientists who played an important role, is Carl Linnaeus, Swedish botanist. He started off this system which contained domains, kingdoms, etc. And Robert Whittaker took this further, and he further modified and developed this classification system, because if you're discovering these organisms, you've got to be able to classify them. His system included 3 domains, 5 kingdoms, and 2 cell types. The domains included eubacteria, archaebacteria, eukarya, etc. The kingdoms included the Monera, Protista, fungi, animalia, and plants. Additionally, as people were discovering organisms, there had to be a system not only to classify but to name them. And this is often referred to as the binomial nomenclature. It usually gives a microbe 2 names, a genus and a species. Genus is usually written with the first letter capital, or first capital letter, and then the second name is usually a smaller letter, and this is italicized. Some common examples include E. coli, Escherichia coli. So Escherichia, the E would be with a capital E, and then you have your coli, both would be italicized. And there's many other examples which you'll come across in microbiology: Staphylococcus aureus, Bacillus subtilis, Bacillus anthracis, Pseudomonas aeruginosa, Klebsiella, etc. Now, within microbiology, there's lots of fields, branches within the microbiology field, and people specialize or do research in different areas, whether it's in basic sciences or in infectious diseases. So if you're planning to become a physician, you could be specializing in bacteria, viruses, fungal pathogens, worms, etc. And if you're in the basic sciences, same way, there's people who specialize in bacterial genetics, bacterial physiology. There's applied fields like medical microbiology, food, agriculture, aquatic, industrial, biotechnology, genetic engineering. Then there's also immunology for those in the basic sciences, and then there's also immunology responses to allergies, etc., allergists, and so forth. Thank you so much for listening to this introductory video, and stay tuned for more videos as we continue this series. Thank you.